Hello, my name is Anthony Moorman and I'm a leukemia researcher based at Newcastle University. And today I've been asked to address the question of why do we have so many different types of acute lymphoblastic leukemia? So this is what I'm going to try to do in the next couple of minutes. Acute lymphoblastic leukemia is a blood cancer that typically arises in the bone marrow of patients. The leukemia arises when a normal bone marrow cell turns rogue and becomes a leukemic cell. And these are often called leukemic blasts. These rogue leukemic blasts divide uncontrollably and prevent normal bone marrow function. This in turn impacts on the patient's immune system and gives rise to the symptoms that they present with, which typically includes recurrent infections. To explain why we have so many different types of acute lymphoblastic leukemia, we need to explore what turns a normal cell into a leukemic cell. And the short answer is genetic mutations. So mutations are changes in the DNA sequence of genes. The genes, which encode proteins, which are molecules that control cell function and also determine whether or not a cell divides or rests, these mutated genes make the cells behave abnormally and turn a normal cell into a leukemic cell. The genetic mutations I'm talking about here are acquired mutations. They are not the kind of mutations that are inherited from parent to child. These genetic mutations or abnormalities are specific to the leukemic cells of patients with acute lymphoblastic leukemia. We don't know the precise number of abnormalities that are needed in order to turn a normal cell into a leukemic cell, but it's probably in the region of 6, 7, or 8. And the key thing here is there are many different types of abnormality that can occur in the leukemia. And there are three main consequences. One is that a gene is turned off and hence there are too much of that protein, too little of that protein is produced. Secondly, a gene can be turned on too high, in which case too much of that protein is produced. Or genes can be reordered, in which case an abnormal protein can be produced. And all these three kinds of abnormalities all contribute to turning a normal cell into a leukemic cell. So in acute lymphoblastic leukemia, the combinations of gene mutations that are present in the leukemic cells define the type of leukemia that the patient has. They determine the different cell functions and also the leukemia cells, they determine the different cell functions that the leukemia cells possess and also how aggressive the leukemia is clinically. In addition, the presence of some of the specific types of abnormality can also help clinicians determine the best treatment option. So far, extensive research has identified probably nearly 100 different genetic abnormalities that contribute to developing acute lymphoblastic leukemia. So the overall whole landscape is quite complex. However, the different combinations of genetic abnormalities are not random. And there are probably around 20 to 25 different genetic subtypes of acute lymphoblastic leukemia, which does sound very high, but the same thing happens in many other types of leukemia and indeed cancer. There are currently many research projects underway, some of them in Newcastle in my research group, looking to see how we can use that information about the different genetic subtypes to inform how patients are best treated and what the optimal therapy is. And they can also be very useful in terms of diagnosing acute lymphoblastic leukemia and also for making treatment options. So that is a very short summary of why there are so many different types of acute lymphoblastic leukemia. As is often the situation these days, the answer lies in the genetic code of the cells that drive this particular type of leukemia. So I'll end there and hope that you enjoy this video, and please do watch more videos on this website. Thank you very much.